will be based off of the those first 4 foundational lectures. So nothing from today is on the quiz for today, just the previous 4 lectures or those modules are on the quiz. Before I dive in, any questions about material from earlier lectures? Good, we're good. Okay, so as I mentioned beginning of the semester, we have a 2-part lecture today. I'm going to describe in very broad terms the process of developing a drug from a laboratory setting into a clinical setting. We have a guest speaker, Dr. Anupam Jigaran from the MSU Innovation Center, who is going to be talking about how you protect an invention, talking about patents, IP, or intellectual property aspects that fit into developing any invention, really, but definitely within the drug discovery process. But he will be talking next. I'm going to do a quick lecture on drug development process, as I just said. And yes, oh, right. And as far as the exam goes, I will post on D2L what will be important from this lecture in terms of the exam on Tuesday, just a few of these slides. Most of this is just more background information about the whole drug discovery and development. So, the term is bench to bedside. It always gets knocked around. This is the colloquialism that we use for actually taking something from the research side, from a university or a company, and developing something at the bench there all the way through to a medicine that is marketed for treating some condition, right? Multiple steps in this pathway, and this is a very broad generalization of them. These steps listed here are the process you will go through, but this is an official list of what you have to do. These are just the steps involved. Of course, you have to start with the discovery. A lot of drugs start off as just an idea, but in many different angles, right? You have a disease state you want to target. You found a protein that you think can treat X disease. You read a paper and you see a structure of a molecule that reminds you of a molecule that treats a different disease, and you want to apply that molecule to another disease. Ideas from all over the place come into this discovery phase, but the idea here is that you hone in on something that you are going to build towards, right? And that happens with all of the next few. So then you develop a set of experiments that you can then test whichever molecule or potential drug you are looking to develop, right? Along the way, you need to validate the experiments or the assays that you create in order to make sure that you have as well or as stringent a process as possible. As you make sure that you want to have a very fit molecule that can advance through the stages of development. Safety issues, monitoring toxicity is something that doesn't happen right away, but it is important in early stages, even preclinical stages. Once you're developing a molecule and you think it might be able to have some potential, you want to start safety studies or toxicology studies as early as possible so that you know you have a molecule that is efficacious but also safe. All of this goes well, you go into clinical trials, we'll get into that in a bit, and if all of that goes well, you can market your drug, all right? So these are, again, very basic steps in this whole process of bench to bedside. Big bottom line here is the cost. Hundreds of millions of dollars to $2 billion, very common estimates for this. I've heard projects $2.6, $2.8 billion to develop. $1 to $2 billion is a very common estimate that's thrown around nowadays for how much it costs to actually develop a drug. So big money, obviously, as well as time. 12 to 15 years. I've seen 10 to 20 years, so this is kind of a conservative estimate, but the bottom line is it takes some time. Real world example is cisplatin, the cancer drug that was invented or discovered here at MSU. That took from the initial accidental discovery to FDA approval was 13 years, all right? And this is back in the 60s and 70s. If you're interested in drug discovery at all and you like good stories, this is an excellent one. This is an amazing kind of accidental discovery that was, again, done here and led to a very successful cancer drug that now has a lot of new variations or generations. But anyways, that's kind of the bottom line with the idea of developing drugs from bench to bedside, all right? So, quick kind of aside, in the field of drug discovery, there really are two main fields, if you will, or main types. Small molecules and biologics, all right? So a small molecule is exactly that. Usually something less than 1,000 Dalton, so Dalton atomic scale for measuring the mass of any molecule. Less than 1,000 Daltons, usually, yeah, a lot of times less than 500 Daltons, but small molecules. Examples would be aspirin, lots of NSAIDs, SSRIs. These are all small molecule drugs, all right? Then there's biologics. Biologics usually take advantage of the hierarchy of molecular biology, right? So you have a gene that's transcribed to a transcript that's translated to a polypeptide or that matures into a protein. Biologics usually take advantage of this because there's a lot of protein-based therapeutics that have really come out in the last 20, 30 years. Examples of those would be antibody-based therapeutics. Those are very big now, and definitely in the field of RNA, really the last 10 years has been explosive in that field for how important RNA molecules are, not just for forming ribosomes and as transcripts for instructing how to create a protein or assemble a protein, but their functionality in the cell is also being used to develop future potential biologics. So, those are kind of the two main fields. Today's talk is mostly going to focus within small molecules. I'll throw in some info about biologics along the way, but for the most part, I'll be just discussing the process of developing a small molecule drug. And here we have, this figure is kind of the entire lecture in a slide, if you will. So the phases that happen, and not going to spend a lot of time on this one. I really just want to call out a couple of things that are of interest, but as you can see here, the stream goes from laboratory, early clinical development, late clinical, and then marketing, right? With the earliest stages being discovery and preclinical. So the discovery side, as we said, you have to hone in on your idea, you have to develop some molecule you want to go after, and then create a lot of assays around that. These are usually biochemical and cell-based assays that you can gain information about the efficacy of potential drug that you're working with, right? All done at the laboratory level. Along with that, with successful completion of enough biochemical or cellular-based assays, you work into a preclinical stage where you're working with actual animals. There are cell-based and tissue-based assays at this level, but you're really focusing on whole organisms, okay? And the idea for getting information, or the requirement for getting a candidate into clinical trials, usually at least 2 species worth of data need to be gathered in order to gain FDA approval to initiate clinical trials. As far as the clinical trials go, traditionally it's phase 1, 2, and 3, okay? More recent stratification of these phases comes here, 1A, 1B, 2A, 2B. Nuances of that aren't so important for our lecture. I want to point out phase 0. This is really a modern thing that's happened, and for good reason. One of the best parts about modern medicine is individualized medicine, is we are understanding how important it is to understand not only genetic backgrounds, but we're finding things that weren't able to be drugged before we're now able to. You know, if you have a disease that has a few dozen people or a few hundred people on the planet, no company is going to touch that. There's no profit in that sort of thing. But now that we have a lot more avenues for philanthropy and funding and whatnot, as well as the clinical space within academic settings is really expanded. And this is very exciting because you can actually test patients with diseases prior to an approval of clinical trial. Now granted, you have to have plenty of preliminary data that shows it's safe and it's efficacious, all right? But you can actually treat patients under a very well-monitored situation, but you can see if it actually has efficacy before initiating a full clinical trial, right? This is kind of a, yeah, this is a great thing, but there's a lot of caveats to it. And yeah. But then there's the 3 phases of clinical trials. I'll get into that later in the talk. And then phase 4, here it's listed as marketing, but you also have a phase 4 that you can do. Usually after successful completion of phase 3 clinical trials, you can apply to have a launch and put the drug on the market. Phase 4 trials are usually for highly specialized situations where if there was some unexpected toxicity that happened after launch and you want to investigate that, you'll form a phase 4 trial. Or what also happens is if you have drug X that treats disease Y, and then you realize that there's potential for drug X to treat disease Z, and you want to get it into that space, rather than having to start all the way at the beginning of phase 1 and everything, you've already demonstrated safety and certain efficacy. This is a way to just run a single clinical trial and have a new approval for that drug with just that input to the FDA, okay? So, let's get back to the beginning here. Drug discovery, initial development, any time frame I have in parentheses throughout this, these are all estimates, right? This is indefinitely 3 to 5 years. People have spent 30 years working on a molecule trying to develop it into a drug. I mean, it's kind of, yeah, it's very drug and individual dependent. Idea here, though, is what are you going after? Did you find a disease you want to treat? Did you find a new protein that you want to drug? Is there a need for a drug in a specific area? Let's say you have, drug X to treat disease Y, but drug X is pretty crappy and it doesn't have real good efficacy. It has shown statistical efficacy, but it's not that great. Is there certain side effects that it's been marketed because it helps the people's lives, but the side effects are horrible? If you could develop something that's better, that already has a drug in that space, a lot of people go for that because there's potential to market that. So, but that's that initial phase of what are you going to do and what are you going to build. From here, again, traditional model, you had in-house specialty research teams, especially in companies. They still have these, but they were a lot bigger prior to this era, where individual companies would have whole teams working on diabetes or hypertrophy or cancer. And those teams would build something from the ground up. We have plenty of this specialty in the research or the academic research phase 2, but we contrast that to the more current model. This is really the last 20-ish years when this has become popular. The acquisition of technology has become a very modern way of doing things. Just due to the fact that there's such a low success rate and you can waste a lot of money, it's better to just find a lab in academia that's developing something and they don't have the means or the resources to push it to the next level. A company can come in and buy that technology from the university and then develop it in their way and then have the money for doing clinical trials. So again, once you have that discovery set and you need to develop it, it used to be done in-house. A lot of companies would do all of the stages from the discovery to the preclinical work through research support groups, process development for building, producing enough of the drug to do clinical trials or the preclinical work. Modern way of doing things are through CROs, or contract research organizations. And these are very big now, as these companies are set up to, they're highly specialized in certain aspects of the drug development game. Okay? And these are excellent, especially for startup companies that don't have a lot of people, they don't have a lot of equipment or expertise, but they have a good molecule that they're working with. They can hire these CROs to do just specific assays and then get the data from that to build their drug off of. Examples here, Charles River is definitely one of the biggest ones local in the Midwest for this type of work. Okay? And, sorry everyone. until recently, we actually, well, I shouldn't say it that way. We have a drug discovery program right here at MSU, all right? MSU Drug Discovery was founded several years ago, looked at as an academic CRO. It's currently in kind of a stalling phase, but there's a couple facilities that are still open. And the reason I bring this up is not only that we have this here, but to introduce the idea of a pipeline. A pipeline are the set of steps that you use in order to develop a drug from just the discovery phase into being ready to file for clinical trials, all right? The steps are shown here on this right column. The idea here is each step in the pathway is designed to really produce the most fit molecule through repeated assays and more stringent interrogation of that molecule. So looking at the steps themselves, target ID and validation, this is that initial step. You know, what do we want to do, what we're going to work with, how are we going to do it, and then assay development goes with that, because you need to build an experiment that you can test the efficacy of your drug. A lot of times, especially in the small molecule world, this is done through high throughput screening. We'll talk about this in the next slide, or a couple slides. Idea here is some people have a receptor or a disease state that they want to develop a drug into, but they don't have an actual molecule. What they do is take libraries of compounds, there's physical libraries that have thousands, even millions of compounds, and they will screen their cells or their receptors with these molecules to see if any of these molecules do to the cells or receptor what they want them to do, all right? Again, I'll get into that in a bit. But from here, you get a bunch of potential molecules that can be used as a potential therapeutic. You continue on successive rounds of assaying and making things more stringent to knock out any lesser performing molecules, if you will. And then you get down to a much smaller group of molecules that are called lead molecules, okay? That's hit to lead. Hits are a big group of molecules, leads are a smaller group of molecules. From there, you do lead optimization. This is where medicinal chemists come in and they can adjust the physical chemical properties of the molecule itself, right? Yes, please.
Let's say if Tylenol, wanted to make extra strength Tylenol, would this process be expedited at all, or would it go through the same stringent kind of like oversight when creating this new drug, even though it could just be like a higher dosage?
Gotcha.
It's already on the market.
So, if you reformulate Tylenol to be extra strength, do you need to do, that's an over-the-counter medication, it didn't go through clinical trials.
No?
Yeah, that's okay, I mean, fair question, but yeah. So medicinal chemists mess with the molecule to make them even better, and then from there, if they perform even better in our in vitro or cell-based assays, go into preclinical testing. And this is usually the flow of the experiment. So you start with your in vitro assays, biochemical assays, you just have an enzyme or a protein or something in solution, and then your drug molecule to see if it does anything. Build from there into a cell-based assay where you're actually growing cells in culture and then bombing them with your molecule with a specific readout to tell you if it's working. And then finally, you build into the in vivo assays, you're actually working in animals for testing your drug there. So, the last thing, but a very important step here is, especially in these last, especially a couple of stages, you have go, no-go decisions, they're called. And this is when you have to pull a plug on, decide whether or not your data is fit enough to continue to advance, or if you want to stop investing in it. As understandably, you would have to be very, these are tough decisions, horrible conversations in rooms, but the idea is, do we want to risk millions of dollars, hundreds of millions of dollars to keep doing this when we're not exactly sure, or how confident are we in going forward? So these are very tough calls that are made during these phases, but that's part of the process. So, when looking at developing small molecule drugs, there is this idea of the funnel that is that happens while doing this. So, as I said, when you're doing screening, especially the high throughput style, you'll take maybe 1 million compounds, okay, and screen all of those for potentials that can work in your disease state or on your receptor or whatnot. You winnow those down to maybe 250 compounds, all right? And then from there, you winnow it further down, and this is a bit misleading here. You usually don't have clinical development for 5 compounds. That would be for this last one here. I feel like this should be up here. You have 1 compound that you go into clinical trials, because again, the cost is really prohibitive to trying to screen a handful of compounds. So you really have 1 molecule that's really important there. Okay, I'm going to go over 2 actual approaches for doing drug discovery and development. This is a very modern technology, but it's getting to be more traditional now, just as due to the fact that it's so well established and has been used for years. So again, the idea of high throughput screening, or HTS. What you do here are using cell-based assays. You will interrogate your molecule in a cell culture and see how well your molecule performs. A lot of times you always need a readout from an assay. A lot of times there are colorimetric or fluorescent molecules that you can use to show success. Specific example in the next slide. But the idea is you do these in these plates. These plates that you can actually grow cells in. So these culture plates are roughly the size of a deck of cards or your phone, really. One of the most common and now earliest renditions of this is a 96 well plate. So there's actually 96 wells. Each of these white circles here represents 1 well that you can grow cells in. Each of the wells will be for a different drug that you're screening. As technology has developed, also the technology of these plates, because then they went to 384 well plates. Most modern version are a 1536 well plate. As you can see, each of these small holes here contains a well that you can actually grow cells in culture and then test them. So each one of those circles will correspond to a specific drug. I've seen people hand pipette these plates before. Yeah, you'll earn ultimate respect if you're able to do it, but most of these are done by robotics now. You're dealing with such small amounts of fluid that you really need a robot to do all of that pipetting, putting in the cells and the media and the drugs and et cetera, et cetera. But bringing this back to the funnel idea, in high throughput screening, you will start with, again, maybe 1 million molecules, even more are very common. And then you perform your assay, and let's say it's a fluorescent-based assay. Let's say you're working on a cancer drug. A cancer drug you would want to kill cancer cells, right? So you have a cell that's developed that when it dies, it emits a fluorescent signal from that. Using green fluorescent protein, if you're familiar with that, is a very common approach. An ideal setting would be on the top. So good bench scientists, you live and die by your controls. The top left here shows positive and negative controls, right? Positive control would be the amount of fluorescence with 100% cell kill. Negative control would be 0 cell kill. And any results that you get are going to be compared to those controls to test their specific fitness. What you'd love to see is a plate with a couple of green spots on it, but what always happens is something like this, where you just get all of this crap in here, and you don't even know, is this green, or what is this? So what you have to do is make judgment calls compared to your controls and select, several of these molecules that then would go on to the next round of screening. You immediately repeat this with these assays, or with this specific assay, and then move on to more and more stringent assays. The last little bit here about nomenclature. So hit molecules, as I said, are the initially successful molecules. So upon first assay, those that look like they're successful are called hit molecules. You winnow that pool down to your lead molecules. So here would be hits, these 5 would be leads, and then your actual candidate molecule would be the one that you're actually advancing into clinical trials. That's your one best bet for success in this game. All right, most modern way of doing things is in silico analysis, really. Modeling software has been used for a number of years, but definitely now artificial intelligence is the big one. And modeling software was developed because we used to be able to, we still can. If you get a structure of a receptor and then you've got drugs and you want to test with that, you upload or you load your structure of your protein into a software and then have it figure out if drugs can fit into an active site or a place where you need it to turn on or turn off a receptor. That was all done in silico, and there was a lot of push and pull in the early years of bioinformatics about whether it works or not. Because you can write a software that says Superman can fly around this room, but that doesn't mean that actually happens. And so a lot of these modeling softwares were generating molecules that when we would test at the bench, they would just fall apart. They've gotten much better, but again, with the development of artificial intelligence, this is really taking over. And here's just one example. So this is from MIT, a couple years old now, but they were using this AlphaFold, which is an AI application, to develop antibacterial compounds. As I'm sure people are aware, antibiotic resistance is a huge headache in the clinical game, and developing molecules just outside of our traditional approaches for killing microbes is very valuable. So here's an example of where they're trying to use an AI-generated model to produce potential candidates. Okay, so everything is successful so far. We've got a lead molecule we believe we can develop into a candidate, but of course we have to make sure that this is going to be safe, right? You can develop a molecule that can cure cancer all day long, but if it destroys your liver, you can't give it to anybody. And these are some of the early tox screens that you'll do. One of the most quick and dirty ways is just to dump a bunch of the drug into mice and see how long they can take it, how long they can process it. Do they get tumors in their liver? Do their kidneys just fall out of their bodies? That is an easy way of testing early toxicity, but through a lot of experience, a number of biochemical assays have been developed with specific ion channels that we know are very important for functioning. Tell a story about hERG channels at the end of this talk. But as we learned before, cytochrome P450 enzymes are the main enzyme in our liver that break down drugs. And does the drug that you're developing inhibit specific CYP enzymes? If you're going into a space where you know people are already going to have multiple drugs on board just due to the condition that they have, is this going to inhibit CYP enzymes so that you cause toxicity, right? Because we don't want drug-drug interactions for a lot of different drugs. Okay, so again, best case scenario, we got all this data, the drug is selective, it can show a therapeutic effect in animals, and it seems to be well tolerated. So at this point, you actually submit all of your package to the FDA and in what is called an IND filing, or an investigational new drug filing, and ask them to approve the initiation of clinical trials, right? They have a specific, short amount of time to act on this and give you thumbs up or thumbs down. All of that nuance is outside of this talk, but that's the process, right? So, on to clinical trials. I show these three just as an example of how infuriating it can be to look up anything in science, because depending on the source, they're going to give you different information. So I won't spend much time here talking about this. The next slides will be the overgeneralized version of clinical trials that I've set up for this class. And the bottom line here is there are multiple phases that have to go through sequentially, and each one has to have a certain success rate in order to initiate the next round, okay? Starting at the beginning, phase 1 clinical trial. This is all about safety, all right? You get a cohort of healthy human volunteers, for the most part, for a short study, all right? You can do different doses, and you can do multiple dosing and escalating amounts. During this phase, plenty of PK/PD studies are done here for evaluating safety and evaluating other aspects of how the drug works. But the endpoint here is safety. How well do people tolerate the drug? The screenshots I have here and on the next few slides are taken directly from the FDA site. Since they initiate these or they monitor these, I figure that's the most reliable information. So, but as this says, phase 1, 20 to 100 healthy volunteers. Caveat here is current cancer drugs, they actually allow cancer patients in the phase 1 trials, okay? But for the most part, phase 1, healthy individuals, small group, short study, several months here, usually up to a year. And yes, you're looking at safety, right? But this is big. About 70% of the drugs make it through this. Pretty good. About 3 out of 4 are successful. That's good. Phase 2, increased by an order of magnitude-ish, several hundred patients. This, depending on how well your institution can gather a cohort to study, can involve multiple institutions. But big thing here, these are placebo-controlled double-blind studies, right? So placebo-controlled, some people get sugar pills, some people get the real drug. Double-blind, neither the patient nor the clinicians know what they're administering until all of the data comes in and they peel off the labels. So nobody knows who's getting what, and some of the people are getting sugar pills. So as the FDA says, several hundred people that have the disease, these are not healthy individuals, or they are currently suffering from the disease. Months to years, 1 to 2 years, pretty common, but here the endpoint is efficacy, okay? However, each clinical trial phase builds on the next, so you're constantly reviewing everything you did in the previous phase, so efficacy and again safety here are monitored for developing the drug. Big bottom line again, you can see about 1/3 of the drugs fall out, or are successful, 2/3 of the drugs fall out. So from the first 2 phases, you're talking 1 in 4 candidates makes it to phase 3. This is a huge setback. Well, tell stories into the end of the night about this, but the bottom line is only about 1 in 4 molecules makes it into that 3rd phase of clinical trials. And I forgot to say that in the beginning. So when I learned clinical trials, it said phase 1 safety, phase 2 efficacy, phase 3 dosing. However, phase 2 and phase 3 really blend a lot of their outputs, if you will. You'll study efficacy and dosing all the way through 2 and 3, but it really depends on the study team, the specific drug, the disease state, all of those things. I've simplified it here just for our purposes. Back to the speech, phase 3, these are hundreds to thousands of people, and usually multiple institutions, just because it's hard to get enough people, but the big development within the clinical trial field in just the last 10 or 15 years, what a lot of companies were doing were farming these out. In Africa and South America, these were really big, because it's very expensive to get thousands of people together to study a disease. However, companies started just going to places, more impoverished nations, because it was cheaper for them. However, they found out there was too narrow of a genetic background to really test not just efficacy, but safety of these drugs. And a lot of companies got in a lot of trouble for this, and they've really done a good job of trying to make much more diverse population for especially a phase 3 trial. Again, we're placebo-controlled and double-blind, and then this is the biggest go, no-go decision here. You have to show efficacy, you really hone in your dosing, and then again evaluate safety. FDA says 300 to 3,000, 1 to 4 years, pretty, can be conservative, but again, big bottom line. Now 1/4 again make it out, right? So even smaller success rate coming out of this, and by this time you're investing hundreds of millions, maybe even $1 billion. So very important decisions, but have to take these very seriously and be honest about how well your drug is working. Okay? So quick aside on what happens during this time too, because it's not just about developing the drug. You also want to continue to monitor safety even outside of this. Is a long-term exposure that goes longer than a clinical trial? Do you develop cancer from it? Do you develop any toxicity? So these will be happening even on the side of this, but on top of that, you have to make sure you can make your molecule. You can have a molecule that kills cancer all day and is safe and it's wonderful, but you just can't make much of it. So you really have to abandon the project, because even if it's amazing, if you don't have enough to supply the market, you're done. On top of that, you also have to prepare for the market. Not only knowing where the patients are and where clinics are that will be treating these patients, but developing sales and marketing team that can get the drug out there, developing the liaisons between the companies that are marketing it and the doctors that will prescribe it, so that they're well versed on what this drug does. All of these things have to happen. So when you get official approval, you're ready to start on day one and get it out there without stall. So, all of this goes well, right? You submit, you got your preclinicals good, 3 phases are good. You know our target population, this whole packet goes to the FDA, and here you file for a new drug application for small molecules or a biologics license agreement for biologics. If that gets the green light, and yeah, here it depends on the urgency that you need. Yeah. If you have a disease that is really affecting, during a pandemic maybe, you can have the FDA fast track their review to get this drug on the market if it looks good. Usually it takes a year or 2, and after you have launched a drug, it takes a lot of monitoring to make sure it's doing what it is supposed to do, as well as there's no hidden toxicities that you didn't see. Okay, so quick aside on, so we just talked about is kind of the traditional pathway, follows these steps, 5 to 10-ish years. All of us just lived through the pandemic, and that required the emergency use authorization, or EUA, of the mRNA-based vaccines. This started in about 2004 when we were meddling in the Middle East, and there was a concern that biowarfare agents were being used against soldiers, so there were a number of agents that were fast-tracked using the EUA to allow for rapid approval. And the way this happens is, rather than just kind of a stair-stepping where you do phase 1, then you do phase 2, you do them all concurrently. Phase 1 and 2 are usually stacked together. As soon as you start seeing promising data from phase 2, you're going to initiate a phase 3 right in there. So you can hopefully shorten this process from years down to months and then get approval of that. So that's part of the game. All right, last thing, I'm going to try to get this out quick. Cisapride is an example of a drug that really caused a lot of problems due to the fact that they missed a certain toxicity during development. Cisapride, 1980 is when Janssen came out, or when they discovered this. So Janssen Therapeutics, subsidiary or part of Johnson & Johnson, drug was approved for treating gastroparesis and dyspepsia. Chronic indigestion. Gastroparesis is when your smooth muscle and your intestine doesn't contract well enough to squeeze that chyme down the tube to form what happens next. So this drug was used to treat that. Launched in 80, pulled off the market in 93. It was discovered in 1980, was launched in 93, pulled off the market in 2000. I'll get into that. Quick background on why. Here's a cardiac action potential. We studied action potential before. We didn't have this involvement of the calcium channel. This is due to where these nerves are being innervated. But remember, a rising phase, sodium in, then we have calcium in, and potassium out. The potassium channel here is called HERG. Stands for human ether agogo, which is a really unfortunate name, but that's the specific calcium channel that acts in these potentials. And as if you can look over here, here's our action potential as it lines up with an actual heartbeat, right? If the clinicians here, you do an EKG or electrocardiogram, you see this heartbeat, right? These are the phases of the heartbeat and how they actually line up to the functioning of an actual action potential. Mutations in this potassium channel cause long QT syndrome, so there's a delay here, and the end of this syndrome, also known as, can develop into torsades de Pointes, is called. But why this is important is cisapride can cause this condition. It's not just genetic mutation, but cisapride can do this, and the reason, or one of the big reasons that it's a problem is because it has a narrow therapeutic index. As we studied before, therapeutic application of cisapride, stomach contractions. Toxic application of cisapride is the HERG inhibition. You can see how close, so here's our dose response curve. The plots for these are very close together, and this drug specifically is broken down by CYP3A4. As mentioned before, this is the most, that cytochrome P450 breaks down most of the drugs that we consume. With a CYP3A4 inhibitor on board, some of them are listed here. A lot of common medications causes a narrowing of that therapeutic index and potential toxicity. And this is what happened. After the drug was marketed, lots of people started having myocardial infarctions. They noticed this, they screened it against this and found that this happens. And now, due to the fact that this drug crashed out and has been pulled off the market, well, for humans, all drugs are screened with a biochemical assay against the hERG potassium channel to make sure that there isn't this sort of phenomena happening. So, I ran long. Quick summary, drug discovery development, lots of years. Pipeline is used for developing the drugs through multiple rounds of experimentation. Understanding the toxicity or potential toxicity of the drug is really just as important as establishing its efficacy. And clinical phase happen in several phases where you evaluate a lot of endpoints, but most specifically, safety, efficacy, and dosing of the drug are focused on. And that's what I wanted to tell you today. So, I'm going to step out and quick introduction. Dr. Anupam Jagran is a senior technology manager here at MSU Innovation, and I'm going to leave it there and let him introduce the subject.
Are you guys able to hear me, or do I get a microphone? Microphone, or hear me?
Good.
Oh, awesome.
Cool.
Thank you, thank you. Thank you all for making some time available and staying back here for this talk. My name is Anupam and I work at MSU's Innovation Office.